Welcome back to this next video in which we are discussing influenza. Okay, so in this next video we're going to begin our discussion of the adaptive immune response to the influenza virus. So we have already discussed the inflammatory response that is going to be initiated against the influenza virus, and that will occur at all the sites where the influenza virus has infected the respiratory epithelium. Now, inflammation is brilliant and it will help to destroy a lot of virus particles. However, on its own, it does not usually manage to destroy every single virus particle, and therefore, on its own, the inflammatory response would lose the fight against influenza. It would certainly slow it down, but it would not obliterate it from the respiratory tract. To obliterate it from the respiratory tract, we need the adaptive immune response to kick in against influenza. So it is important that we're now going to discuss this because this is how you fight the influenza virus off. Okay, so let me just say a few words about the spirit with which we're going to discuss the adaptive immune response. So I could spend hours and hours and hours describing the adaptive immune response, and indeed in another video entitled The Immune System in the playlist on immunology, I have spent hours and hours and hours going through the adaptive immune response. It's a 24-part video, I believe, and in that video we go into a huge amount of detail of the adaptive immune response. I'm not going to go into that level of detail here. I'm going to give you the bare skeleton of the adaptive immune response. And in a way, I quite like the bare skeleton of the adaptive immune response because a lot of people, when they study the adaptive immune response, they end up learning all the little details of the adaptive immune response, and they end up with this absolutely massive amount of information that they're supposed to learn, and they learn it all in little chunks, and then they struggle to put all of the little chunks together to get the bigger picture of what actually happens in the adaptive immune response, with the consequence that they know all the little details. They know, for instance, the in-detailed signaling pathway of how B cells are activated and the in-detailed signaling pathway of how T cells are activated, but then they don't understand the bigger picture of the adaptive immune response, of how the whole system works together to actually obliterate pathogens. So actually, it's quite nice to just discuss the skeleton of the adaptive immune response because you really do understand the big picture here, whereas when you study it in full detail, you can, to use the old cliche, lose sight of the wood for looking at the trees. So, that's the spirit with which we're going to do this. We're just going to do the bare skeleton of the adaptive immune response, and this will allow us to see the big picture of what is happening, but I will not be going into the details of exactly how it works. If you want the details of how the adaptive immune response works, as I say, I refer you to the other video entitled The Immune System. There we go into it in full detail, the full detail that I know. Okay, right. So, next thing to say is that the adaptive immune response is very different from the innate immune response. It is specific to the pathogen that it is initiated against. The innate immune response, the cells of that and the proteins of that were completely non-specific. They were not directed against any pathogen in particular. Macrophages and neutrophils just go around looking for any old pathogen, and they do the same thing to them all. They try and break them down through phagocytosis. Natural killer cells go around looking for cells that are misbehaving and have too low MHC class 1 on their surface. Those mechanisms are not specific at all to the pathogen that we are dealing with. And the proteins, the antimicrobial proteins in the exudate, they're certainly not specific to the pathogen that they're dealing with. They're non-specific as well. Whereas in the adaptive immune response, we are going to pick out special cells within the body that are specifically targeted against antigens of the pathogen that we are fighting. We're going to make them divide and divide and divide, so we end up with loads of these cells that are all directed against the specific antigen of the pathogen that we are fighting, and then we're going to unleash them all on the pathogen, and they will attack it and destroy it, and that is the spirit of the adaptive immune response, that the troops that are going to fight the pathogen will be specifically designed against that pathogen, whereas the troops of the innate immune response were not specifically designed against the pathogen. And this is why the adaptive immune response takes time to initiate, because you have to find the special troops that are directed against the pathogen that you're dealing with, and then you have to get them to multiply, you have to clonally expand them, and then you're ready to actually start attacking the pathogen. Okay, so the adaptive immune response, there are multiple different types of adaptive immune response. There are multiple different arms of the adaptive immune response, and in response to broadly different types of pathogen, we initiate different types of adaptive immune response. So for instance, in response to fungi, there is a certain type of adaptive immune response that you initiate called the T helper 17 response. That's a more niche one. In response to influenza, the two most famous arms of the adaptive immune response are going to be initiated. That's why this is good revision of our immunology, because we're going to get to discuss the two most famous pathways of the adaptive immune response. So let me just tell you about what those two are, or remind you, hopefully, about what those two are. So the most famous, most iconic one of them all is the B-cell response, and influenza is going to have a B-cell response initiated against it. So the B-cell response is one of the arms of the adaptive immune response, the major arm of the adaptive immune response, certainly the most famous, and it's going to be a pathway that we're going to use against the influenza virus. Now, the B-cell response, of course, has another name. It's also known as the humoral adaptive immune response. Now, why is it called that? Well, remember, the way B cells actually attack the pathogen is they produce antibody molecules, they produce immunoglobulin molecules that will go into the body fluids, which used to be called the humor of the body. Humor refers to body fluids. So these molecules will go into the body fluids, and then they will be the specific molecules that attack the antigens of the pathogen. So that's why we call this the humoral adaptive immune response, because the troops are in the humor. They are protein molecules within the humor. Okay, so that's one of the major arms of the adaptive immune response that we will be discussing here. Then the second most important, most famous arm of the adaptive immune response, which again is going to be initiated in response to influenza viruses, is the cytotoxic T lymphocyte or cytotoxic T cell response. So cytotoxic, which just means toxic to cells, and then it's the cytotoxic T lymphocyte or cytotoxic T cell response. And for short, you can abbreviate cytotoxic T lymphocyte down to CTL, like so. So cytotoxic T lymphocyte response. Now, the cytotoxic T lymphocyte response is an example of what we would call a cell-mediated response of the adaptive immune system. Now, that means that the troops that are going to be produced in this cytotoxic T lymphocyte response are going to be white blood cells. They're going to be cytotoxic T lymphocytes, as opposed to molecules, proteins that are in the humor. And there are other examples of cell-mediated adaptive immune responses that aren't going to be important in the influenza virus, such as the T helper 17 response, the T helper 1 response is another example. We'll mention them later on as we go through our discussion of these two pathways. Okay, so we are going to initiate these two separate pathways of the adaptive immune response then. Now, let me just give a bit of motivation about these pathways. What sort of pathogens do we launch a B-cell response against, and what sort of pathogens do we launch a cytotoxic T lymphocyte response against? Well, think about this one firstly. We produce antibodies in the B-cell response. Antibodies can bind onto the specific antigens on the surface of the pathogen, such as, for instance, we could produce antibodies against hemagglutinin and neuraminidase. So they could bind onto those, and they're actually extremely useful for all sorts of things, for attacking that pathogen. And we'll come on to how they attack the pathogen later on. But they do attack the pathogen, and they do lead to the pathogens being made useless and being destroyed. So, which sort of pathogens do we want to initiate that against, and which sort of pathogens would it not be so worthwhile initiating a B-cell response against? Well, any pathogen that is extracellular, that is going to exist in the extracellular space, it is worth initiating a B-cell response against. So that is the key thing that we need to understand about the B-cell response. You initiate this in response to pathogens that are extracellular. And the reason is that antibody molecules are going to be in the extracellular fluid, and they cannot go into the inside of cells, antibody molecules. So if you've got a pathogen that spends all of its time inside cells, and there are some very clever pathogens which say, let's avoid the B-cell response, let's avoid antibodies by staying inside cells all the time, and will spread to other cells by getting the cell that we're currently in to go very, very close to the other cell, and then we can just hop between the cells. So they never actually go into the extracellular fluid. There are examples of pathogens like that never go into the extracellular fluid. So if you do have a clever pathogen that stays inside cells all the time, there is absolutely no point initiating a B-cell response against it, because the antibody molecules will not be able to get at that pathogen. So, you initiate a B-cell adaptive immune response against pathogens that spend at least some of their time outside of cells. Now, influenza virus is an intracellular parasite, of course. It goes inside cells and that uses them to make loads of copies of itself, but it also spends time outside the cells. And whilst it's outside the cells, it will be vulnerable to antibody attack, and therefore it is worthwhile to initiate a B-cell response against influenza virus. So remember that. That's a key lesson. B-cell responses, if you've got an extracellular pathogen, even if it doesn't spend all of its time extracellularly, if it spends any of its time extracellularly, we will initiate a B-cell response against it nearly always, because it's always going to be helpful. Okay. Cytotoxic T lymphocyte response is less famous than B-cell response, because obviously this one's major. A huge number of pathogens have to spend some time outside of the cells. Cytotoxic T lymphocyte response is less famous because it's initiated in response to less pathogens. This is the extracellular response, this is the intracellular response. If we just initiated this one, then we'd be able to get, loads of the influenza viruses whilst they're outside of the cells. But what about the cells that actually have influenza viruses inside them? We need to be able to kill the intracellular influenza viruses as well. This response can only kill the extracellular ones. So when you have a pathogen that is intracellular at some point in its life cycle, we do need another type of adaptive immune response to deal with that, and that is what this cytotoxic T lymphocyte response is for. It is the way of killing intracellular pathogens, and the way that it works is that we produce cytotoxic T lymphocytes that go around, find virally infected cells, and kill them, just like the natural killer cells do, but these ones are more powerful at doing it than natural killer cells. Natural killer cells rely on the cell having too little MHC class 1. This one can see much more clearly that the cell is infected with a virus. It gets an absolutely clear signal that the cell is infected by a virus, whereas the natural killer cells get a more ambiguous signal. They have to go up to cells, try and work out how many MHC class 1s they have, and then they have to have few enough MHC class 1s that it's a significant enough drop for this cell to actually be activated to kill the cell. So you do actually have to have a big drop in MHC class 1 in order for the natural killer cells to be confident enough to say that they want to kill the cell. And of course that's perfectly reasonable, because we don't want natural killer cells that will go around and kill any cell that has a few too little MHC class 1 molecules on its surface. That would be no good at all. It will go around killing those innocent cells. For that reason, natural killer cells have to have quite a high threshold. It has to be a massive drop in MHC class 1 for it to be sure that this cell is dodgy and to unleash its killing mechanisms on it. So the point is that natural killer cells are very, very tentative about killing virally infected cells. It has to have lost a huge number of MHC class 1 before they will then say, okay, I'm confident will kill this cell. Whereas the cytotoxic T lymphocytes, they don't need as much stimulation at all. They get a very clear signal that instantly tells them this cell is virally infected, and then they say, we'll kill it. So they have a signal that they're much more confident in, and therefore they kill a lot more virally infected cells, because a lot more of the virally infected cells will give them a signal that they can be confident enough to kill the cell compared to the natural killer cells. Okay, so I hope that what I've just said is clear. So these are very, very good at killing virally infected cells. So this cytotoxic T lymphocyte response is the way that we get rid of intracellular pathogens. So obviously, in response to influenza, we need to initiate both a B cell response to get rid of the extracellular influenza virus particles, and we also need to initiate the cytotoxic T lymphocyte response to get rid of the intracellular influenza virus pathogens. Okay. So, those are the two responses that we're going to initiate. We understand the motivation behind these responses, one being extracellular and one being intracellular. Now, let's actually go to the technicalities of how this happens. So, it starts with T-cell activation. Both of these pathways start with T-cell activation. This one, it's obvious that we're going to have to activate T-cells. This one, it's less obvious that we're going to have to activate T-cells. However, in order to activate B-cells, you have to activate special types of T-cells called T-helper cells. So I'm going to start with T-cell activation. We will discuss the cytotoxic T lymphocyte response, and we'll finish that one, and then we'll talk then about B-cell activation, and we'll finish the B-cell response. So it'll all be clear right at the end. Just go along with it if you're not too clear about immunology at the moment. You'll see how it all fits together. So we need to activate T-cells then, and T-cells are all directed against specific antigens of pathogens. We have a huge number of T-cells in our body, and we have a huge number of B-cells in our body actually as well. And they reside in the blood, they reside in lymph nodes, they reside in the spleen. And, you know, there are billions of these cells in the body, a huge, huge number of different T cells and B cells. And they all are directed against different antigens. They're specifically not just directed against an antigen, they're directed against a little fragment of antigens, a little peptide fragment, and they all have different little peptide fragments that they're directed against. Now, when we get infected by influenza virus, we therefore need to chop up its antigens into little fragments and take these fragments to the T cells, and we need to try and find T cells that are directed against those fragments and activate them. And that's what's going to happen in the initial part of this response. So as I say, I'm going to do the skeleton of this. So the whole thing begins with antigen-presenting cells. So we'll draw a little flow diagram for this. So antigen-presenting cells are going to begin the whole thing. APCs for short. So this stands for antigen, that's the A. And we can abbreviate antigen, and I will abbreviate antigen in future down to AG. Remember, an antigen is any protein that can have an immune response initiated against it, an adaptive immune response initiated against it. All the proteins of influenza virus, not just hemagglutinin and the neuraminidase, the other proteins as well, M1 proteins, M2 ion channels, non-structural protein 2, all of them, they're all antigens. They can all have adaptive immune responses initiated against them. So A is for antigen, P is for presenting, and C is for cells. So these are cells that are going to activate T cells. They're going to activate the adaptive immune response. And all of the different arms of the adaptive immune response always involve T cells. So T cells are the central cells of the adaptive immune response. It's only this one arm of the adaptive immune response, the B cell response, that involves B cells. Every single different type of adaptive immune response always involves T cells. They're the central cells, or T lymphocytes, of course, they're the same thing. Lymphocyte, T cell, you know, you can use either name. Okay, so what are the antigen-presenting cells then? Well, we have met them already, thankfully. Dendritic cells and macrophages are the antigen-presenting cells. So there are two different types of antigen-presenting cells, the dendritic cells and the macrophages, which were also the sentinel cells. Now, remember, we discussed that when you get an inflammatory response in an area, you're going to bring in loads more dendritic cells and macrophages, particularly macrophages, but remember, you'll also be bringing in dendritic cells. When the monocytes come out of the blood, I told you that they could differentiate into both dendritic cells and macrophages, and some of them will differentiate into dendritic cells, and not all of them will differentiate into macrophages. So the point is that in the inflammatory response, you bring in loads of dendritic cells and loads of macrophages into that underlying connective tissue that of the respiratory epithelium, and of course then they can go into the actual respiratory epithelium itself. Now, both of these types of cells are phagocytes. We talked about macrophages long ago when we did inflammation. So down, where is it? Here. Okay, so we talked about macrophages a lot when we did the inflammatory response. We didn't talk about dendritic cells as much, but they are phagocytes as well. I didn't talk about dendritic cells as much because they're not as much part of the inflammatory response. I mean, we bring in loads of macrophages, we don't bring in as many dendritic cells, but they are now important for initiating the adaptive immune response, i.e. the change between the inflammatory response, the innate immune response, to the adaptive immune response, which is why I'm now starting to talk about them. So, not only are neutrophils and macrophages phagocytes, but also dendritic cells are phagocytes. They can phagocytose pathogen particles, and now I've got to try and find, here it is, the adaptive immune response. And so they can phagocytose pathogen particles, and then they can unleash their lytic enzymes on those pathogen particles. They will break down all of the proteins of the pathogen down into little fragments. And what can now happen is these antigen-presenting cells, the dendritic cells and the macrophages, can put the fragments of the antigens of the pathogen on their surface attached to major histocompatibility complex proteins, and they can present them to T cells. Now, at this point, there is an important difference between what happens to initiate the B cell response, the extracellular response, and what happens to initiate the intracellular response. So let's have a look at this. So we're drawing a flow diagram here. So the dendritic cells and the macrophages, cells that we've met before, these are going to be the antigen-presenting cells. They're going to phagocytose the pathogen. However, if they're not just going to phagocytose the pathogen, if they phagocytose the pathogen, so I might just draw a little picture of this. So let's draw it for a dendritic cell. So we'll have a dendritic cell here that has maybe come in as part of the inflammatory response and is now, let's say, going to be exposed to an influenza virus particle. So here is an influenza virus particle. What it can now do is phagocytose that and break down all its proteins and start presenting them on major histocompatibility complexes. If it does that, think what this cell knows about this pathogen. If it has phagocytosed the pathogen directly, that means that the pathogen was in the extracellular fluid. So this antigen-presenting cell knows that this is an extracellular pathogen. So what type of adaptive immune response is this antigen-presenting cell going to want to now initiate? It's going to want to initiate the B cell response. So this is important. The decision of which adaptive immune response we are going to initiate is starting to be taken already by the antigen-presenting cells here. If they find an extracellular pathogen, they decide that they want to initiate a B cell response, an extracellular response, rather than an intracellular response. So, whoops, I want the pen back. So, when they actually phagocytose the pathogen directly, which I might just write, it's the influenza virus, but of course this goes for more general pathogens, not just the influenza virus. So if they phagocytose the influenza virus directly, they are going to want to initiate the extracellular response. So it's going to go down the extracellular response now. So it cuts up all the antigens of the influenza virus into little fragments, and it's now going to put those fragments on its surface. Now, if it wants to initiate an extracellular adaptive immune response, i.e. the B-cell response, it needs to put the fragments on a certain type of major histocompatibility complex, and that one is MHC class 2. So, I'll stick to the red pen here. So, it chops up the pathogens' proteins, and then it's going to present them on its surface. So I'll just put a little bit of membrane of the dendritic cell here. So this can represent one of the dendrites of the dendritic cell. What's now going to happen is I'll get a new colour. We'll have green here. On these special proteins on its surface, and here it is in green, this is an MHC class 2, and this stands for Major Histocompatibility Complex class 2. Now, I've already written in this video Major Histocompatibility Complex, so I'm not going to force myself to rewrite that out again. I'm sure you can remember how to spell that. But this is not the one that we discussed earlier. We discussed the Major Histocompatibility Complex class 1 earlier. This is a different one, a very different one. Initiates the completely different adaptive immune response to MHC class 1. MHC class 1 is the intracellular response. MHC class 2 is the extracellular response. So, if the antigen-presenting cell, either a dendritic cell or a macrophage, finds a pathogen in the extracellular fluid, phagocytoses that pathogen, it knows this pathogen spends time extracellularly. It says, I want to initiate a B-cell response. I want to go forth and activate T cells that are going to help initiate a extracellular response, a B-cell response. And to do that, I have to put the fragments of the antigens that I have got of this pathogen on MHC class 2 molecules. So, it cuts up all the proteins of the influenza virus in random ways, produces loads of fragments, and each one is going to put on an MHC class 2 molecule, as I will show here. So this is the MHC class 2. As you can see, it has got this nice little space here, which is representing a groove in the protein molecule, which is a complex, which is called the peptide binding groove. So major histocompatibility complex molecules have a little groove on their surface called peptide binding groove, the peptide binding groove. And that is what is represented there. And this is where they can bind a little peptide. And this peptide will be reasonably short. It'll be between 10 and 25 amino acids. Okay, so it puts a little peptide there on its MHC class 2. And it won't just do this for one peptide. It will do this for loads of peptides. It's got a huge number of peptides from cutting up the proteins of the influenza virus. So all over its surface, it will now put MHC class 2 molecules with different peptide fragments in the peptide binding groove. Now, at the same time, there won't just be one antigen-presenting cell doing this. There'll be loads of other dendritic cells all over the respiratory tract and loads of other macrophages all over the respiratory tract that will also be phagocytosing influenza viruses, cutting up all the proteins into little fragments and sticking them all over their surface on MHC class 2 molecules. So you will get loads of antigen-presenting cells covered in MHC class 2 molecules with different peptide fragments of the influenza virus antigens in their peptide binding grooves. And the key thing that I want to stress from that is that different antigen-presenting cells will cut up the antigens in different ways, and therefore will end up with a whole different set of peptide fragments from the same set of antigens of the influenza virus. Because remember, they cut the polypeptides up in this random way, so where they put the cuts is going to be continuously different. But the point is that because so many antigen-presenting cells are doing this, you're going to get a huge number of the different possible peptide fragments that it is possible to create from the influenza virus antigens actually being presented to T cells to try and activate adaptive immune responses. So you'll get a huge host of different peptide fragments that are all parts of antigens of influenza virus. Because you might have been wondering, how does this cell decide how to cut up the antigens? Well, it just does it randomly, and all the different antigen-presenting cells will do it in different ways, therefore, but overall, because so many of them are doing it, you will get big scope of the different possible peptide fragments that it is possible for you to create by chopping up influenza viral antigens. Okay, so I hope you understand that. So, leaving off on the extracellular response for a moment, let's talk about what will happen with the intracellular response. But just before I say that, let me say that the major type of antigen-presenting cell is actually the dendritic cell. Macrophages will do it to a degree, but dendritic cells absolutely love to do antigen presentation and activate T cells. This is their major time to shine. They may not come into the inflamed area in the same level as the macrophages do, but they have a task to do, and they, by God, do they do that task. They find pathogens, phagocytose them, and then go off and present the peptide fragments from their antigens to T cells. Whereas macrophages, some of them might do that if they feel like it, but a lot of them don't bother. Okay, now, that's how we're going to start the initiation of the extracellular response. Antigen-presenting cells are also going to start the initiation of the intracellular response. So how are they going to do this? This involves them phagocytosing something different. Think now. These antigen-presenting cells, if they want to decide that they're going to initiate the intracellular response, they need to somehow know that the pathogen is intracellular. How might they know that the pathogen is intracellular? What have I told you gets phagocytosed by macrophages and neutrophils and actually also gets phagocytosed by dendritic cells, even though I didn't tell you that at the time? The dying epithelial cells. Epithelial cells that will be full of influenza viral antigens. And that, if one of these antigen-presenting cells was to phagocytose that, then it would know that the pathogen was intracellular and would then start the process to initiate an intracellular response. So that's exactly what is going to happen. So you see how beautiful the adaptive immune response is. It does make sense. And so, let me draw here in white. Here is one of these ciliated columnar epithelial cells, and let's say this is dying because it is infected by influenza viruses. And I know, of course, the virus particles won't be assembled inside the cell, but just to show this, I have put a particle in there. In fact, that's so wrong, I will change the picture. We'll just put a cross for it to say that this cell is dying because it is infected with influenza and it is full of influenza viral antigens. So what's going to happen is this is going to be phagocytosed. I'll get another colour now to do the opposite pathway, so we'll go into this mint green colour. So what's going to happen is when you phagocytose dying cells, so the other option is that you can phagocytose a dying epithelial cell that is infected with influenza virus particles. And then, of course, the antigen-presenting cell will be aware that the pathogen, the antigens that it's getting and chopping up from this pathogen are now intracellular. It will be aware that the pathogen is intracellular. So again, what it's going to do when it phagocytoses this is it will break down all of the proteins here, and the antigens of the influenza virus that are inside this cell will all be cut up into little fragments as well. And now they are going to be put on MHC class 1 instead to initiate an intracellular response, because this antigen-presenting cell knows now that these antigens are associated with an intracellular parasite because it found them inside another human cell that it was phagocytosing because that human cell was dying. Excuse me. So, what it's going to do is it's going to cut up all the influenza virus antigens, and it's now going to try and initiate an intracellular adaptive immune response, i.e. a cytotoxic T lymphocyte response. So the intracellular one. And to do this, it's now going to put the peptide fragments from the antigens that it has cut up on its surface attached to MHC class 1 instead. And I think actually I'll just leave a little bit of space there because otherwise I'm not going to have anywhere to go with this arm of the flow diagram. So I'll go downwards here rather than along there. Okay, so let's draw this on then. So here again is just representing the surface of a dendritic cell. And now let's have a different colour for MHC class 1. Let's go for, what should I do? We'll have purple. So here this is an MHC class 1 molecule, and it's the same principle as MHC class 2. These again have a peptide binding groove where they can put a little peptide from some antigen that has been chopped up. So what's now going to happen then is peptide fragments from the influenza viral antigens that were found inside the cell will now be put on MHC class 1 molecules. Okay, so this is what antigen-presenting cells do overall. To summarise what we've seen so far, antigen-presenting cells are going to activate the adaptive immune response, all of the different arms of the adaptive immune response. The two major types of antigen-presenting cells are the dendritic cells and the macrophages, particularly the dendritic cells. They love doing this. Macrophages, some of them will do this, but most of them won't. Now, dendritic cells then, well, not just the dendritic cells, antigen-presenting cells, they actually take right at this point the decision of whether we are going to initiate an intracellular or an extracellular adaptive immune response. In response to influenza, we know that we need both of them initiated. So, when an antigen-presenting cell phagocytoses the pathogen, which is just floating around in the extracellular fluid, it knows that that pathogen is extracellular, so it says, let's initiate an extracellular response, and to do that, it chops up its antigens and presents the peptide fragments of those antigens on MHC class 2 molecules on its surface. When you want to initiate an intracellular response, why would you want to do that? Only if you find it inside cells. So how are they going to find it inside cells? Well, they will phagocytose dying epithelial cells that are full of influenza viral antigens. They'll break down all the proteins of that cell, and that means that they'll also break down all the antigens of the influenza virus. They'll cut them up into little fragments. They then, because they know that this is now intracellular pathogen, you know, the antigens they found here are an intracellular pathogen, not an extracellular pathogen. They then want to initiate the intracellular response, and to do this, they put the fragments on MHC class 1 molecules on their surface. And again, of course, they'll have cut up loads of antigens. They'll end up with a huge number of peptide fragments, so they will be coated with MHC class 1 protein molecules with these different peptide fragments in their peptide binding grooves. And again, when the different antigen-presenting cells are cutting up different, influenza viral antigens, they will cut them up in different ways so that you get a huge variety of different peptide fragments. So you get a huge scope of the different possible peptide fragments that you could get by cutting up influenza viral antigens. Okay, so this is antigen presentation. So what are these antigen presenting cells going to do now? Well, of course, what do they want to do? They want to activate T cells. T cells are the central hub of the adaptive immune response. So where are all the T cells? Mainly in lymph nodes. So what will now happen is the antigen-presenting cells will move into lymphatic drainage vessels in the tissue that they are present. So, of course, we're talking about the respiratory tract. So the dendritic cells and the macrophages will drain in lymphatic vessels that drain the respiratory tract to local lymph nodes. And there they will come into contact with T cells. So what's now going to happen is the antigen-presenting cells will go to lymph nodes, and there they're going to activate T cells. So again, the T cells that we're going to activate are going to be very different. So we've reconverged the pathways here to say that they're both at this point going to now go off to lymph nodes. However, we're going to very quickly diverge the pathways again. So, I think we'll discuss firstly the extracellular pathway, and then we'll come back to the intracellular pathway, and we can finish the intracellular pathway very quickly, whereas for the extracellular pathway it's going to take quite a bit more, because of course we're going to have to discuss B cells. They are the central cells of the B cell response, but we're not going to see them yet. Okay, so we go off to lymph nodes then, so let's do the extracellular response. What's going to happen? So we'll have another colour. I'll pick a different green, I think, a very bright green. So we are doing then the extracellular response here. So I'll just put EC again for extracellular. So I've used EC for endothelial cell, epithelial cell, and now extracellular. Okay, so we're going to activate T cells then, and the type of T cell that we're going to activate in the extracellular response is known as CD4 positive T cells. So the type of T cells we're going to be activating are CD4 positive T cells, or CD4 positive T lymphocytes. Now, to just draw a little picture, T cells are very boring cells. They're spherical, like so, and they have a very large nucleus. So here is the nucleus of our CD4 positive T cell. Okay, now you will have in your body a huge number of CD4 positive T cells, a huge great population of CD4 positive T cells, and they like to hang out in lymph nodes. A few of them will be in the blood, and some of them will be in the spleen, and they move around the body. They don't just sit in a lymph node for your entire life, sitting there bored stiff. They do move around. They say, I'll hang around in this lymph node for a while, then I'll go back into the blood, I'll go to another lymph node somewhere far, far away, then I'll go to another lymph node, etc. They move around. They don't stay in one lymph node for their entire life. Okay, and different CD4 positive T cells are different. They are directed against different peptide fragments. So all the T cells in your body, not just CD4 positive T cells, but the other population of T cells as well that are directed against, that are going to be important in the intracellular response, all of them have a structure on their surface called the T cell receptor, which I'll just put here. So in vivid purple here, this is the T cell receptor, and for short the T cell receptor is abbreviated down to the TCR. And each of the different CD4 positive T cells has its own unique design of T cell receptor. And what can the T cell receptor do? It can bind to MHC molecules, major histocompatibility class molecules, when they have peptide fragments in. Now, each T cell receptor will be designed to bind to either MHC class 1 or MHC class 2. All of the CD4 positive T cells, their T cell receptors only bind to MHC class 2, not MHC class 1, which is why they're going to be involved in this extracellular response. But all of the different CD4 positive T cells, they will have slightly different T cell receptors that can recognize MHC class 2 with a different peptide fragment in. So I hope you understand this. All of the CD4 positive T cells, they are directed against certain peptide fragments. They all have a peptide fragment that they absolutely hate and which they are programmed against. And if this peptide fragment is presented to them by an antigen-presenting cell in an MHC class 2 molecule, so if this peptide fragment that they hate is put in an MHC class 2 molecule and the antigen-presenting cell brings it to them, that is going to activate them. So, all of the different CD4 positive T cells have different peptide fragments that they are directed against, and this is how the adaptive immune response is going to become specific. So, let's do this then. So remember, we're only doing the basic skeleton. I'm not going to go through the details of how CD4 positive T cells are activated. If we've had the influenza virus phagocytosed from the extracellular fluid, and we've cut up its antigens into little fragments, and we've presented them all in the peptide fragment binding groove of MHC class 2 molecules on the surface of our antigen-presenting cell, and it's now come to lymph nodes, and what's going to happen is it might well come into contact with CD4 positive T cells that have a T cell receptor that is directed against the peptide fragment that they are presenting. And of course, they will have a huge number of different peptide fragments, and the different antigen-presenting cells will have different types of peptide fragments because they've cut up the antigens in different ways. So what's going to happen is, because we've got so many different peptide fragments being presented to T cells in the lymph nodes, eventually we're going to get some hits. Some of the peptide fragments are actually going to find a CD4 positive T cell that's directed against them. And when that happens, what's going to happen is the CD4 positive T cell is going to divide and divide and divide and divide. So they get activated when an antigen-presenting cell actually presents to them the peptide fragment that they are targeted against. And as I say, we're going over the bare skeleton of the adaptive immune response, so we're not going into the details of how they become activated. they become activated, and of course the response to that is clonal expansion. This CD4 positive T cell is going to be useful for us. It is directed against a peptide fragment of one of the antigens of the influenza virus. It's going to be useful for getting rid of the influenza virus. So we make it divide so that we have loads of them. So let me show this here. So division is going to occur. Here's 1, here's 2, here's 3, 4, I'll draw a few more, 5, 6. Of course, division to a much bigger scale than this is going to occur. You're going to get a huge number of these CD4 positive T cells that are all identical and which are all now targeted against this peptide fragment. Then what's going to happen is they're going to start differentiating. However, I think we will have a break here and we'll continue this story in the next video.